Welcome back to Educator.com. This is Laboratory Investigation 3, Carbon Dioxide Production. Alright, so with the CO2 lab, the purpose is to measure the difference in CO2 production in humans before and after exercise. When I do this with students in a classroom, it's very interesting, their reaction to this, because they go into it just thinking that this is a fun lab where I exhale through a straw into the solution. It's fun. We're all doing it for a minute straight, and we're going to have this color change. It's going to be great. And then they find out they have to go exercise during part of it. And they're like, man, I didn't think this was going to be PE, physical education. But you know what? Biology, study of life, it's important to engage in physical exercise. So that's a crucial part of this lab in terms of measuring how much CO2 is exiting the body before and after exercise. So the material, 2 beakers, 1 large, 1 small. Here's an array of beakers. You could use this one and that one. You could use this one and that one. It really doesn't matter the exact size as long as they're all above 50 milliliters. So just kind of medium-sized beakers will do. You're going to put 50 milliliters of distilled water into the larger one, and you're going to put 20 milliliters of 1 molar dilute concentration, or even more dilute, of sodium hydroxide, NaOH. You don't actually need a separate beaker for the 10 milliliters, or you could even use less. You could probably get by with just a few milliliters of hydrochloric acid and this phenolphthalein. The hydrochloric acid, you're going to use just a few drops midway through the lab. The phenolphthalein is what's known as a base indicator, and I'm going to tell you more about that later on in the explanation. A pipette, you probably would need 2 pipettes or medicine droppers because you're going to be putting a base, an alkaline solution, drop by drop into one of the beakers, and you're also going to put HCl, an acid, into that same beaker eventually, so that you don't have to clean the pipette every time between going from acid to base. You don't want to contaminate the inside of the pipette and affect what you're putting in at what time. So having 1 pipette for basic solutions and 1 pipette or medicine dropper for acidic is probably a wise idea. Drinking straw. You're not actually going to be drinking out of it, but you'll be exhaling through it. And then a clock or watch, a way that you can measure the time. You just need it for 1 minute. Time required, about 30 minutes is all you need. Part 1. All right, of course, before you begin, you got to make sure you're using gloves and goggles, safety first, because even though these are very dilute substances, if they splash on your eyes, in your mouth, if you get them on your hands and then put your hands in your mouth, it's not a good thing. So make sure that you are protecting yourself if you were to actually replicate this lab, and do it with the supervision of a teacher or professor. So part 1, put water in the larger beaker, NaOH or sodium hydroxide in the smaller one. You're going to use the drinking straw, which you're not actually going to drink out of, to exhale into the water, the beaker with the water, for 1 minute straight with every breath. Now here's what I mean. So when the time starts, you're going to have your little beaker there. Okay, go. So if you notice, I was inhaling through my nose and exhaling through my mouth. So that's what I mean by every breath. Make sure that every breath, you're not actually exhaling through your nose, you're exhaling through your mouth. And part of the inhaling through the nose is just to prevent you from inhaling any liquid. So part of that is do not submerge the straw into the liquid. Now for this first part, in the beaker, there's just distilled water, okay? Now, if the beaker's not very clean, there could be some remnants of other chemicals from previous labs. Also, the reason why I'm saying not to submerge the straw is at this time you might only accidentally inhale some water, but later on in this lab there's going to be other chemicals in that beaker. There's going to be hydrochloric acid, there's going to be sodium hydroxide, phenolphthalein, things you do not want to swallow and get into your body. So don't actually dip the straw in the water, just hover it over the liquid. So after you have exhaled all that CO2 into the water, you're going to add 1 drop of that phenolphthalein to the water. Phenolphthalein is a base indicator, meaning if a solution is on the alkaline part or basic part of the pH scale, it's going to turn this color, this pinkish magenta color. If it's acidic, it'll actually be clear in this lab. So once you get past the 7 on the pH scale, you're going to see it turn this color. And the more base you add, it will get darker and darker, kind of to like a reddish, even kind of light purple eventually, if it gets really, really basic. So that's going to come in handy in terms of measuring a change with a visual indicator in this lab. So the 1 drop of phenolphthalein is all you need. If you put too much, the color change will happen too quick for our purposes. So just 1 drop's fine. You're going to put NaOH, which is a base, into the water drop by drop until it becomes like this, until it turns to this color. This is your end point that you're shooting for. So maybe with that 1 molar, that particular concentration of NaOH, of sodium hydroxide, maybe it's just 3 drops. That's all you need to get here. Or maybe it's 6 in this lab. It's going to be relative to the amount of acidity that's in the liquid. And if this is not really making sense yet, you're going to see at the end of the lab when we analyze what's happened here, I'll kind of draw it out for you in terms of what happened. So you want to record the amount of drops of NaOH that it took to do this, whether it was 3 drops of NaOH, or maybe it was 7 drops of NaOH. Really depends.